So welcome to our panel on variation in regulatory approaches across the globe and how we can work together to make medical products commercially viable and available worldwide. So variation in the way regulatory agencies view medications or devices can teach us about the outcomes in different countries, and this natural variation can give us the opportunity to harmonize when that is appropriate, or learn from each other, or develop better approaches by working together. And we have an example of each one of those issues on this panel. So everyone's aware of the problems of getting drug approvals harmonized, and that is being worked on. However, there are a number of other important regulatory issues that we would also benefit from learning from, and how we could do a better job of harmonizing. So we have 4 examples, and we're going to try and get through this in time. So our first example is one about pancreatic islet cell transplantation, a super interesting issue that we're going to, that Peter Stock from UCSF, transplant surgeon, is going to start with. I'm joined by Tina Morrison in moderating this panel. As you heard, she is the director of the CERSI program at FDA. And we're not going to go through the bios. You can read about them in the program or ask ChatGPT. Okay. So these are our panels, our islet cells, drug or transplant. And we have Peter Stock, Thierry, Berney, James Shapiro. Then we're going to go to product lifecycle for devices and strategies to accelerate progress. Then we're going to talk about adapting corneal grading systems to assess unexpected toxicity, eye toxicity, in new oncologic products. And then touch on the universal need for better data generation. Okay, Peter, you're on.
Thanks, Laura, and thanks for the opportunity to join this panel. Pancreatic islet transplantation for the treatment of diabetes is a perfect example of how we might be able to learn the most successful global strategies for regulation, with very, very different regulations in the United States versus, in fact, the rest of the world. Just as a brief background, transplantation of beta cells, either as a whole organ pancreas or as islet cells, both result in states of insulin independence, both require immunosuppression, and both are derived from deceased donor organs. Can you go back to the last slide, please? In 2016, we reported that both the costs and the outcomes of as it relates to insulin independence, we're getting closer for both solid organ transplantation and islet transplantation, prompting this cover of the American Journal of Transplantation addressing which one of these therapies is going to win out, the whole organ pancreas, the islet cells, or the computer in the background representing closed-loop devices for insulin delivery. Next slide. So pancreas transplantation is performed about 1,000 times a year in the United States, usually in conjunction with a kidney transplant. It is a very rigorous procedure, a lot of cardiovascular stressors for the patients, so that the vast majority of people with diabetes can't benefit from that operation. Now, the regulation of solid organ pancreas transplants is under the auspices of HRSA and the OPTN/UNOS. And that's the case for all solid organs that we transplant. Of note, we're transplanting this whole pancreas, which consists largely of digestive enzymes, for the 5% of beta cells that are contained within the pancreatic islets. So next slide. Islet transplantation is a safer, simpler procedure. We start with the same pancreas that we use for whole organ pancreas transplantation. It's placed in a digestive chamber with collagenase. The enzyme is activated by heating up the circulation, and gradually the pancreatic tissue dissolves. The islets are released from the tissue. Next slide. And this is what it looks like. The islets are stained red with diphthosome. The rest is digestive tissue. Next slide. And then those are purified on a density gradient. Next slide. And then those islets are percutaneously injected into the portal system of the liver by our interventional radiologist, essentially a same-day procedure. The islets diffuse out into the periphery of the liver, and there they can create states of insulin independence. Now, islet transplantation is regulated by the FDA as a biologic drug. So with this degree of manipulation that I talked about, it makes sense that this needs to be regulated by the FDA. On the other hand, islets are in essence mini organs containing a network of vessels, nerves, and multiple endocrine-producing cells. So it also makes sense that maybe this should be regulated by the same structure that's in place for solid organ transplantation via HRSA and the OPTN. Now this is, can I have the next slide, Laura? This has led to very large disparities in the way islet transplantation is regulated across the world. In the United States, islets are regulated as drugs since they do not meet the exemption criteria for drug regulations. Specifically, as per the FDA, islets are for allogeneic use and therefore should be regulated as drugs, and islets are more than minimally manipulated, so should also be regulated as a drug. Now in Europe, Canada, Australia, and Japan, allo islets are regulated as a tissue or an organ since they do meet the exemption criteria for drug regulations. In Europe, it doesn't matter that the islets are used allogenically, as separate transplant regulations in place for all the other solid organ transplantations reassure safety of allogeneic use. And also in Europe, they state that the biologic characteristics are not changed during the production, and therefore they don't need to be regulated as drugs. So the consequences in the United States, allo-islet transplantation is not available at the almost every transplant center, and only one private company affiliated with an academic institution got a license, a BLA, but in Europe, allo-islet transplantation has been available to patients in transplant centers, and safety and efficacy has been verified in over 1,000 patients. Next slide. So I'm going to turn this over to our next two panelists who will address the regulatory structure of islet transplantation in their countries. Both are transplant surgeons who understand that successful islet transplantation will likely put us all out of business, but that's okay. We're looking forward to that. Our first speaker is James Shapiro, professor of surgery at the University of Edmonton. And James really kick-started the whole field with this landmark publication in the New England Journal of Medicine in 2000. He'll speak first, and he'll be followed by Thierry Berney, Emeritus Professor of Surgery at the University of Geneva and director of one of the most successful islet consortiums in Europe. Now, Thierry pointed out, can I have the next slide? Can you go back one, Laura? You missed one. Skipped one. Yeah, we had there was supposed to be a slide in there that was a picture of the world.
It's right here.
Oh, there you go. So despite the fact that the early innovation in the United States and reports of comparable success in 2016, islet transplantation, as reported by Dr. Berney, has fallen off the map in 2023 in the United States. So I'll turn it over to James.
Okay. Joining us online, if you can show our two panels online. James, can you comment on the strategy that you've taken in Edmonton?
Thank you very much. Thanks, Peter, and thank you for the privilege of the floor. Well, not a lot has changed since we first met with the FDA at a meeting in 1999 with Dan Solomon chairing that, other than accumulation of a lot more safety data. We're all aware of the FDA's motto, trust in God, all others must provide data. And in 2024, we now have a lot more data, published, validated safety data with islet cell transplantation. Pancreas and islet processing is nothing like a drug. The biggest variable is still the quality and differences between human donor organs, not the final product that's released for the patients. The final product QC release criteria clearly work and are safe based on, in Edmonton, 765 intraportal islet cell transplants carried out over the past 24 years. We've never had a liver abscess. We've never transmitted a tumor. We have no uncontrolled or unregulated hypoglycemia. The risk of the cells not working, primary non-function, is exceedingly low, about 0.13%. Donor CMV transmission is the only infectious risk that we've ever observed and can be mitigated by sustained CMV prophylaxis in the recipients where there's a mismatch. We've transplanted 765 intraportal islet transplants into 328 patients to date. Health Canada approved islet cell transplantation on the 1st of April 2001. That was 10 months after the New England Journal paper was first published, and has continued to approve, similar to other solid organ transplant regulation, with GMP manufacturing of islets since that time. So the FDA lags 23 years behind Canada on this issue, and as a result, Americans with severe labile diabetes are missing out on access to this therapy. A 10-center NIH ITN trial and then the two regulation BLA trials, CIT-06 and CIT-07, were completed with over $100 million spent of NIH funds, and they continued to demonstrate the safety of this cell-based therapy. 20 years later, we published our outcomes last year in Lancet Diabetes. I won't go into all of the details of that paper, you can read it, but we essentially showed profound and sustained protection from severe hypoglycemia, improvement in hemoglobin A1C, and periods of sustained insulin independence with improved quality of life for patients receiving islet cell transplant. Human islet transplants with minimal manipulation are clearly not drugs, and the risk to recipients is clearly low, provided GMP manufacturing and final product release criteria are adhered to. And I think still we don't have the regulation for this quite right in the US. Thank you.
Thank you. Thank you for your point of view and all of your data. Okay, so Thierry. There we go.
So, yes, I just had to unmute myself. So thank you for the invitation to share the European experience and most specifically the experience in Switzerland and France. So just as a background, regulations for these kinds of issues are made in Europe by the European Medicines Agency, or EMA, and although not part of the European Union, Switzerland also complies with these rules. So EMA has ruled already in 2011 that islets were not what is called in Europe an ATMP, Advanced Medicinal Therapeutic Product, which is the equivalent of what is called a biologic drug in the USA. And this was emphasized by the following 2 points. First, that islet manufacturing and conditioning does not involve substantial manipulation, and there is a whole lot of actions on the tissue that is listed, and to emphasize that it is not substantial manipulation. Also, islets do not divide or proliferate when they are put in an incubator, and they are used for the same essential physiological function in the recipients as in the donors. And this was the major reason why it was classified as not as an ATMP, but as a cell or tissue transplant. Also was emphasized the fact that the tissue from which islets are manufactured is based, and I quote, on a philosophy of voluntary and unpaid donation, altruism of the donor, and solidarity between donor and recipient. And that was thought to make this procedure not compatible with the for-profit manufacturing and marketing of the islet product. As Dr. Shapiro has said, it cannot be overemphasized that islets are clearly not a drug. There is donor variability, which is the major reason for this, but it's not the only one. And at the moment, there is no real-time reliable potency test that can, which is what is required for a drug. And unlike for a drug, not a single islet preparation is identical to the other because of what I've just said. So what about the situation of islet transplantation in Europe, but mostly in Switzerland and France? So there have been a lot of prospective and retrospective trials conducted in Switzerland and France between 1999 and 2020, including 2 completed prospective randomized control trials, which, as Dr. Shapiro has said, have also demonstrated the safety and the efficacy of islet transplantation in over 800 transplants performed in Switzerland and France, primarily really in Geneva and in the French Brazil network. So based on these observations and also on the US publications, we put forward a file to the authorities, and the Swiss Federal Office of Public Health has included islet transplantation in the list of reimbursed therapies in 2010, and the French Haute Autorité de Santé, the High Authority for Health, in 2020. And in both countries, islet transplantation is considered as standard of care and reimbursed modality for patients with complicated type 1 diabetes. An important thing also is that because of that, islet transplantation is managed by the national organ sharing agencies in both countries, and pancreases are allocated for pancreas or islet transplantation according to organ transplantation rules. And the final point that I would like to make is that islet transplantation has demonstrated survival benefits in candidates for islet after kidney or simultaneous islet kidney transplantation. And there are a lot of patients who are unable to undergo whole pancreas transplant, mainly because they have cardiovascular issues that make them not fit for a pancreas transplant. And in this respect, and this is something that was argued in a viewpoint paper recently by myself, the lack of access to islet transplantation could be seen as some kind of discrimination when these patients are compared to the patients who are fit for pancreas transplantation. So thank you again for the privilege of the podium, and I'm really interested to see how this debate will unfold today. So thank you very much.
So thank you so much. And, so this international variation, I would say, certainly can provide new feedback. So Peter Marks is in the audience. I wondered if you would be willing to talk about, has this information that's been generated, could that be helpful in maybe being repackaged to maybe get this issue, reconsidered in the U.S.? Is this...
Right. So thanks very much. I think this is a great example of yes, but. So I think you're right. There's a lot to be said here for why this is a very important procedure, and we think we acknowledge that. But let's just be very clear. By our laws in the United States, our regulations, pancreatic islet transplantation is a drug, okay? That's just how we define a drug, which is it's more than minimally manipulated, and an allogeneic product, it's a drug. And that said, there probably are ways that we can do it better. We've come up with ways of doing it better, for instance, in terms of simplifying the situation for cord blood cell transplantation. So it may be that there are ways that we can help get to the right place where there's a reduction in the regulatory burden. I just also want to point out something that's also, just with all due respect, Switzerland, Canada, and the European Union, those countries have different healthcare systems with centers of excellence where if we have a little bit of a different system here in the United States. So I think, would like to work together to find ways to decrease regulatory burden, but to get to a place where we can ensure that the products that will get to people will be safe. So I think there is some compromise here in the middle.
Well, that's super helpful. And as we are committed to making progress, I think everyone would be willing to work together. Everyone, yes? Peter, Thierry, and James, to try and think about this. I think one of the most compelling things is that if you regulate it like a transplant, that puts some quality control in. I don't know how you do it hybrid, but we will report back next year about what happens. This is a great opportunity to revisit. So I hope I have, raise your hand, you guys, if you are interested in working on this. Yes, Peter, is a yes to you. Thierry, James? Yeah, great.
No pressure.
No pressure. Okay, awesome. Thank you so much. Okay, Great, okay.
Okay, so moving on to other examples where there are variations in regulatory processes across the globe and how that can impact organizations and companies. We have another example of highlighting the challenges for companies to align their timelines for their products to launch, for their approval across different regulatory bodies. There is a forum, the International Medical Device Regulators Forum, where regulators do come together to talk about some of these topics, and here to share an example of that is Dr. Doug Kelly from FDA.
All right.
Thank you very much. Can you hear me? Great. Next slide, please. So when I came to the FDA 3 years ago, kind of when the FDA was getting swamped by COVID, and part of the reason I came was because for 30 years I was a venture capitalist and funded a lot of early stage, seed and early stage med tech companies, biotech companies, laboratory instrumentation companies. And the med tech realm is a very unique ecosystem. And I thought I'd outline some of these with this slide I put together, kind of difference between biopharma and med tech. And the basic one is that once a biotech drug gets its FDA approval, that's basically the major risk inflection point. And then everything post-FDA is pretty transparent. That is not the case for med tech. Med tech has payers that you have to go through. You have to go through physician professional societies. You have to go through appeal to patient advocacy groups, hospital purchasing managers, contractors, all kinds of other things. That is largely opaque. And that is the reason that you can take a very early stage biotech company public with minimal data, and you can't take a med tech company public because there's still so much risk involved. And that's why mergers and acquisitions are small in med tech as opposed to biopharma. It has all these knock-on effects, this siloization, this opacity of the post-FDA process. So when I came to FDA, one of the things I really wanted to work on was how do we get everybody to work together to create a much more transparent and much more parallel process than exists now. So if you've got a PMA product, the most rigorously regulated products, that could take you 20 years from product ideation to profitability. And there's no fund that sits around for 20 years waiting to fund these things. There's many other more attractive opportunities that are much shorter in time frame. It's really not geared towards a venture model. So that's some of the things that we're hoping to work on. Next slide, please. So here's an example. This is the typical McKinsey chevronogram of what happens from an idea to ultimately to patient access. And our goal is timely patient access to safe and effective medical devices. And you see here you've got idea, the FDA part of it, and then all this other stuff downstream. And the problem is when these things fail, oftentimes the founders of the companies have no idea. If I asked anybody, if I took a show of hands, how many people here understand the CPT process? How many people can tell me what RUC means? I mean, it's very different. I mean, there's a million things like that on the medical device side. Even venture capitalists don't know it or understand it. So what's clear is that there's a lack of toeholds, there's a lack of relationships. It's not for want of trying, it's just that nobody really knows how to do it. And there's never really been leadership or funding in the federal government to really try to pull all this together. And that changed with Jeff Shuren. Under Jeff Shuren's leadership, he's a true visionary at CDRH. So the goal is to keep this cycle of failure. When those things fail, the companies the founders that don't get to go start a new company, even if they were right, that their product was actually really good. And so there's this kind of deflation of the innovation cycle. Next slide, please. So again, we have these stakeholders, the FDA, providers, in terms of professional societies, payers, and patients. They just don't know how to talk to one another. Next slide. And the problem is you're trying to develop a kind of a complete data set that would be convincing to bring to the table as an advocate that you had enough evidence that really showed you solved the problem. If you don't talk to anybody, the odds of you having a critical overlap of this Venn diagram is not going to happen. And that's what happens right now, which is why the failure rate is so high. It's this lack of early coordination of stakeholders. So what we try to do is bring everybody together in this new thing that we have called the TAP program. We talk to bring innovators in early. We want to be the first stop. We are the first stop that says to innovators, prove that what you do works. And in the course of doing that, we want to hear input from what do physicians think about this? What does the leadership of the professional society think? What do patients think? Are these endpoints that they think are interesting? Oftentimes they're not. And what do payers think? That payers have big pain points. They're a business. And to think that you can have a product that ignores what payers want is crazy. So the idea is, again, listen to everybody, come to a consensus about what evidence you can, that they're asking for, and you as an innovator, you're the only one who can decide how much risk you're willing to take. And that's actually another thing, is that when the risk is all bottled up in things outside of innovation, you don't have any room at the end to really innovate, which is why so much in medtech is different catheters. There's very little really breakthrough stuff at FDA for medical devices, because so much of the risk tolerance is taken up by what do I got to do do my payer study? What do I got to do to get my CPT code? And how am I going to fund the company for all that? Whereas you can see this crazy stuff in crazy innovation, fantastic innovation going on in on the biotech side, CRISPR and CAR-T and everything else. So here's an example of what we're doing. We're hoping that by bringing everybody together and having a lot of conversations, more informal conversations very early in the process, and bringing everybody in, because the problem is if you're a payer and you expect that passively you're going to get what you want just by somebody throwing something over the transom, it's not going to happen. So what we're hoping is by engaging with people and getting them out of this passive role, that somehow maybe through a miracle of God something will be thrown over at them that makes healthcare much more affordable and much better. They have a stake in the solution. Next slide. So this is a group that we put together called TAP. It's like a startup inside of FDA. We're kind of a skunk works for FDA process. And we bring everybody together in this kind of virtuous cycle of evidence gathering. Next. So again, conclusions. Early, continuous, interactive. FDA innovative stakeholder communication, more risk capacity to devote to really clinically relevant product innovation. Helping innovators understand. 70% of the people that come to us at CDRH have never been to the FDA before. It's their first time. So it's not all big companies. It's mostly small companies. And we want to help them develop products that actually meet a real need so that we can achieve our mission, which is timelier patient access to better characterized and therefore safer and more effective medical devices. Thank you.
So one of our goals was to definitely try to get audience participation in this panel. As you know, with this very contracted time, we're thinking we had a targeted comment from Josh McAuer from the Stanford Biodesign to comment on. The Stanford Biodesign works with novel innovative groups and investors in the Bay Area on these products. If you have a comment to share with the community on this, thank you.
Sure, thank you very much, and I just want to congratulate Doug on a very innovative and important program, which I hope is valuable and delivers on the objectives. I think everything that Doug said is exactly accurate. We have huge challenges on innovation in the United States, certainly, with respect to the reimbursement phase of and execution of adoption. And that does generate more costs and makes patient access very difficult. So I think that all of these efforts are incredibly important to make it better. And in the spirit of the entire panel, I think this is there have been tremendous progress that Jeff Shuren and Doug have executed on that have actually put forward first-in-man studies here in the United States and other innovative opportunities for young startups here in the United States that really have accelerated innovation in a very positive way on the regulatory side. And interestingly, at the same time, while let's say 12 years or 13 years ago, prior to the FDAsia, we saw that Europe was way ahead on innovation. And I think that because of all the changes that Congress enacted and also the implementation here in the United States with CDRH, we've seen major steps forward in terms of support for innovation with FDA's partnership. And I'd love to see the same type of collaboration on the reimbursement side. So I applaud what Doug is doing. A lot more work to do and a lot of opportunity to better patient access and outcomes. Thank you very much.
Thanks.
Thank you.
Can I add a comment to that?
Yeah, please.
So, payers, CMS, private payers, everybody else, one of their concerns with innovation is that this is just swamp the system, that will just be taken over by a lot of medical devices that who knows what it does or whatever. When you look at the two most risky parts of medtech development, it is really, at the end of the day, the biggest one is actually fielding your sales force. You have to field a sales force. If you're a small company, it means you got to hire a bunch of people who are unproductive for the first 9 months, and that's often a bet the company kind of thing. So when you have a breakthrough product, most breakthrough products fail. They're completely bleeding edge. they don't make it through their clinical study. They just don't show that it works. So the ones that do then have to raise money to build a sales force. A lot of them can't do that. And then when they get that sales force, it has to be effective. So that's the other thing. And so if you actually look at the exposure, the economic exposure to more fully covering breakthrough medical devices, it's actually a nit. Because at the end of the day, those companies that survive to make those products is actually quite small. So the actual overall exposure to really support innovation like was proposed with MSIT is actually very small. And it's something that really people don't talk about. They just assume every single device that goes through the breakthrough program gets funded and it's on the market, and that's not the case.
Well, we certainly would rather venture money go into breakthrough devices than dog food, but I mean, personally. But I would say, I mean, it's going to go into something, but if you can't make the process better, it's going to fail.
Well,
so
it
I
just
think
takes
this
too long.
could really help with biomarkers, liquid biopsies. This process of getting everybody together first and say, you know, I sit on the medical advisory panel for Blue Shield, and like most of the studies that are done, you know, the panel's like, well, they haven't proved anything. The studies weren't done in a way that will get the panel to approve it. And it's like, then sit down with these people at the very beginning and find out what the bar is and how to design the trials, because then they can't get the approval, which then they can't get the funding, I mean, the CPT code or the other funding, right?
Yeah, and that's actually one of the issues with the TSET program, is
Yeah.
that they don't talk to you until you've already done your study. Well, would have been better to talk to me before I spent the $100 million or
Yeah.
$500 million on my clinical study.
All right,
At the
Doug, well,
beginning.
so we're expecting you to have a couple of great examples of how this succeeded.
I'll solve it
by
all
next
this year.
Okay,
All done.
fantastic, okay. All right, I want to introduce a third case, and this was really a very opportunistic thing. New antibody drug conjugates have been introduced, and there was an unexpected toxicity, which was keratitis. And as the chair of my Data Safety Monitoring Board for the I-SPY trial said, oh, ophthalmology and cancer, that's it. That's the death knell for this drug. Why don't you just take it out of the trial? But I had sat next to Neel Pasricha at my fellow's wedding. And I called him up and I said, Neel, I need you to solve a problem. So, Neel, tell us how you solved the problem.
Problem solved. So thanks, Laura, for having me, and thanks for everyone at CERSI for putting together such a great meeting. So as Laura mentioned, I'm a cornea specialist, and so I was told that there's only 4 ophthalmologists here. So for everyone who's not an ophthalmologist, the cornea is the very front part of the eye. And so I focus on that 0.5 millimeter that's right at the front there. So next slide. So antibody drug conjugates, like Laura just described, are a relatively new class of cancer drugs that are very targeted. So it has an antibody that's conjugated to a toxic payload by a chemical linker, and it's great. There's 11 that are FDA approved. There's over 150 that are currently in clinical trials. It's a great targeted therapy, but unexpectedly, they cause these microcyst-like epithelial changes in the cornea. And so I've zoomed in on the cornea in very high magnification. You see those tiny little dots that are there next to the light. Those are the problems. And what happens with these microcyst-like epithelial changes is you get pain, you get blurry vision, and it's actually, in some of the drugs cases, it's the most common adverse event for some of these ADCs. So what we found is that when Laura told me about this problem, and when I saw the grading scale, I said, oh my God, this looks terrible. Like, this looks very confusing, and as an ophthalmologist, I don't know what is a grade 1 or 2. It kind of mixes visual acuity with some clinical changes. And what we were finding is that a lot of the patients that are in these ADC trials fell under this grade 2 corneal keratitis. And that was about 50% of our patients. And what was being recommended is that those patients need to have a dose delay in their medication. And in some cases, these ADC drugs were life-saving, and they were having them delayed because they had corneal changes that were reversible. And so we identified 3 major problems with this. The first is that these scales are confusing. They mix visual acuity with clinical findings. The second is that they don't have photos. We in ophthalmology love photos. We're very visual. And then the third is that there were patients who were getting unnecessary dose delays.
Which also meant the drug would be dropped. You couldn't study it, really.
Exactly. And so what we decided to do is put together this multi-center interspecialty consensus working group to solve this problem. So we had oncologists led by Laura, ophthalmologists led by myself, and then we also had the FDA members led by Wiley Chambers. And we worked together to redesign the scales to make sense, and not only to be easy to understand, but also have the correct oncology dose recommendations. And so what we were left with then is this beautiful new updated grading scales that were very easy to understand. And so, for example, this is one that would be for the cornea. And you can see there, right off the bat, it has nice pictures, it looks nice, it's easy to understand from an ophthalmologist standpoint, and has very clear, easy to understand recommendations on the oncology side. And so this kind of had three different things that we liked. The first is that it had the very clear, easy to understand language, had the photos, and that the dosing recommendations were appropriate. And we're excited to have these rolled out, not just for antibody drug conjugates, but potentially even for other oncology drugs that are going to cause ophthalmology issues. So thanks again, Laura, for getting me involved.
Well, and Neel, you're also trying to solve the problem of access to ophthalmologists, which not so easy to get an appointment all the time. So you're working on automating this with the iPhone or some device like that, right? So you can have a virtual ophthalmology consult, right?
Exactly.
Okay, and could we turn to Wiley Chambers? So Wiley, this is, I think this is an example of how you helped we rapidly solve this problem. How can we share this, or how can we now get the data, again, used and shared with agencies around the world as these drugs go forward?
So thank you for having me. The CTCAE, which is a common terminology for adverse events used by oncology, has been in place for a long time, but it didn't work for ophthalmology. And it didn't work because frequently the time to intervene, as Neel was just talking about, you get adverse events that are not necessarily noticed at the time that you need to be intervening, and you don't have the right match. So up until now, we had basically just said, don't use the CTCAE for ophthalmology events. And so just basically pushing off the whole problem. As pointed out, pushing off the problem doesn't always work. So we went to the next step. Okay, let's see if we can find a scale that fits into the CTCAE so you can replace what was being done with a system that could then be shared. And the CTCAE already is used widely. It just didn't have the right set of terms for an ophthalmology problem. And so this was a way of putting something in the CTCAE that can then be used for any oncology trials, as well as other trials worldwide that deal with the severity of this corneal problem. Now, I think there are two issues. One is the severity, which we fixed. The other is the causality. And while this fixes the severity, it doesn't fix the causality. And I can't tell you how many times I've heard somebody tell me, well, I thought the event was not related to the drug. This ocular event was not related to the drug because I've never seen it before with this drug. Well, of course you haven't seen it before with this drug because the drug is being studied for the first time. So that was never a good way. Asking clinicians to try and come up with what the causality of something they've never seen before just doesn't help. So I think that my preference for any of these, certainly for the ocular events, is just to record what events you have and then look and see if there's an imbalance between the events. And if there truly is an imbalance, then it probably is being caused by the drug. And if it's the same thing uniformly, then it's probably just by background of the disease. But I think fixing this with PTA go ahead.
Wiley, it's such a sensible approach. Shouldn't this just be what we are doing for all adverse events and saying, instead of asking physicians to ask about adjudication, get the series of things that you want to report, the ones of special interest, new ones that come up, and just look for imbalance and just record it?
I think this would save a whole lot of time and effort on the part of the clinicians, and it's certainly within our division frequently ignored So why are we spending time having somebody try and figure it out when we're going to, at the agency, said certainly for ophthalmology, we're going to ignore it?
Well, fantastic. I think that's a great advance. That would be a great advance. So speaking of that, let's talk about how we can change evidence generation in the first place.
Yeah, so lots of innovative things coming out of FDA, as you hear from our FDA colleagues, so keep that in mind. Our last example is really trying to address a common problem many of our physicians face in the capturing of source information in clinical trials. It's expensive, it's time-consuming, and many of them are replicating the effort. So here to share a little bit about that is Ali Abbasi. Thank you.
Thank you, Tina, and thank you, Laura, for putting on such an incredible panel. And also thank you to the other panelists. I think now we know that if someone passes out, it's not because their eyelid transplant is malfunctioning, it's just because we're late for lunch. So I'll try and keep it brief. So I want to try and tie this whole panel together and talk about a common challenge in regulatory science. And this goes to, I think, what the heart of the issue is for a lot of the challenges that we've just heard about, which is that we still struggle to generate evidence at the point of care that is fit for purpose for both regulatory considerations and post-market evidence generation and surveillance. And as the commissioner said in his remarks earlier, it's easy to make decisions when the evidence is clear, but unfortunately, the evidence is usually far from clear. And we've all been talking about the learning health system now for about 15 years, and we're still a long way away from that vision that was outlined in the initial report. So how does this relate to what we just heard about? So let's start with the safety issues that we just heard about in the ophthalmology realm. A large part of safety surveillance in the US for both pre-market trials and post-market is based on case reports and attribution. This notion that we see an event, we fill out a form, and then we ask, could this be related? Now, that's not a really useful way of looking at safety, especially, as Wiley Chambers mentioned, if we've never seen that agent before. The problem is that in our healthcare system, writ large, we struggle to generate the denominator. The way to look for an imbalance is to look for the number of events that occur amongst people that take a product compared to some control group. And ideally, you'd get some percentage. But because we don't collect systematically data on everybody, we have no idea what the denominator is, and that leaves us, both FDA as a regulator and trial sponsors, drowning in adverse event reports with very limited ability to weed through them and figure out what's actually related to the trials. Similarly with the islet cells, Peter Marks mentioned, we need to be sure that when these products hit the market, they will be safe. And again, our lack of ability to surveil safety, product quality, and efficacy in the real-world setting is a major limitation to our ability to bring these products to the market in the first place. And then going to Doug's example, medical product developers spend a lot of time and effort on developing these products. And a major barrier to bringing new products to market is the time that it takes to conduct these trials. So if we could do that faster, more efficiently, that would open up a whole new realm of medical product development. And the consequences of this failure of evidence generation are severe. As the commissioner mentions frequently in his remarks, life expectancy in the US is dropping for the first time in recent memory. Providers are reporting record levels of burnout, in part related to the burden of documentation, of entering the same information several times, but not getting anything for that. As Laura likes to say, she spends, as I can attest painfully, she never gets home from a clinic day before 9 or 10 in the evening. But what do we get for all that documentation? We can't ask Epic, how many patients with HER2-positive breast cancer did we see today? We can't ask Epic, how many patients on a study drug had an adverse event compared to patients on another drug. So we're not getting anything for all the effort we're putting into collecting that information in the EHR. So if you look at the next slide, I think there are some solutions that are being developed. First of all, we have to be clear what the target is. The target is to improve the experience of patients and providers, and in the process, accelerate the pace of innovation and improve evidence generation. Now, there's some progress already happening, and some of this is led by the FDA. We heard last year about improving access to clinical trials for diverse communities, both domestically and abroad. There's been huge progress made in the realm of pragmatic trials, in part due to the pandemic, with examples like the recovery trial in the UK, the active trials, FDA's Project Pragmatica, and there's lots of ongoing efforts led by the commissioner at the FDA to accelerate the conduct of streamlined evidence generation. There's also some areas where there's opportunities, and we need to make sure to harness them. And I'm talking specifically about the rise of AI. Now, there's been a lot of discussions about the power of AI to change healthcare. And I think we need to make sure that as AI becomes prevalent, we direct that towards something that makes the experience of patients and providers better and accelerates the pace of innovation to get better products to market. What do I mean by that? Now, if you look at what AI is being used for currently, specifically large language models, there's a lot of talk about having large language models write your note for you in the clinic. Now, that's great. That might make you more efficient. But as a provider, the last thing that I want is more free text. Why are we asking these advanced algorithms to essentially do what we ask an intern to do, to create more free text notes? When I show up in pre-op the morning of a surgery, I don't want to read the whole story from the pre-op note. I want some key information, like what is the tumor stage? What is the nodal status? What lines of therapy has that patient received? And I think we need to think about ways that we can use these new tools, like large language models, to capture that information in a systematic way and enable downstream uses, not just to make clinical care better, but to accelerate research, surveillance, public health surveillance, even billing can be made easier doing that. And the final thing I want to talk about are incentives. Now, as all of you are highly incentivized to get to lunch, I imagine there won't be any questions after this panel. And so you'll be able to empathize that incentives really drive how people behave in the real world. And the reality is that financial incentives in our healthcare system, not just in the US, but globally, are not aligned to generate evidence at the point of care about what works. What are the hospitals going to do when a large language model writes the notes for the physicians? Well, they're going to say, wow, you're so much more efficient now. Why don't we cram another 3 patients into your clinic day? How can we develop incentives that harness that technology to make the experience better for everyone, but also generate the evidence at the point of care to figure out what works and overcome all these challenges in regulatory science that we've heard about? And with that, I think I want to thank everybody again for your time.
We can get that done in the next year, too. Right, Ali? But this is the next generation, and we know it's going to get solved. So there you go. Big hand for all our panelists.